So, what are blood parasites? Which live in the blood, either intracellular or in the amidst the blood cells or in the plasma. The most common is the Plasmodium species. There are four species: the uh, Plasmodium malariae ovale, then falciparum, and the vivax. The disease is it is a digenetic parasite, and the human being as well as Anopheles female Anopheles mosquito, they are responsible for the completion of the life cycle of this disease. It is transmitted by the female Anopheles, and the characteristics of this disease is the periodic attack of chills, high fever, and profuse sweating. Now, they belong to a phylum Apicomplexa, as you know, have already mentioned that they have a very complex means by which they attach for the completion or for the penetration within the host tissue. Now, regarding the history, it has a remarkable history. In the 1880, Laveran, he was awarded the Nobel Prize when, due to his contribution by finding the parasites in the blood of a malaria patient. So he established that the parasite is present in the blood of the patient. Then subsequently, Camillo Golgi also established that there are two forms of the disease, and these two forms are related to the periodically periodically reappearance of the fever. So that is called tertian periodicity, where fever every 48 hours, and one with quatern periodicity with every 72 hours. That means after every third day, fourth day, the fever appears. And tertian every third day. After 2 days, third day, the fever appears. And Camillo Golgi also, his contribution is that he studied, he gave the conclusion that the fever, the appearance of the fever, it coincided with the rupture and release of the asexual stages of the parasite into the bloodstream, and that is responsible for the onset of fever. And subsequently, we are aware of the um, Sir Ronald Ross, and he was a in the Indian medical service. He was the first to demonstrate that the parasite actually resides or dwells in the lining of the stomach of the female Anopheles mosquito, for which this breaking discovery he was awarded Nobel Prize in 1902. Now, regarding the importance all over globally, this disease is very much it causes lots of life. Fatality is in increase. In India, also in 2021, the number, 161,753 malaria cases has been reported, with subsequent number of deaths also. Now, coming to the infective stage, the malaria parasite, it is just a plasmodium sporozoid. Okay, it is a plasmodium sporozoid. Now, the structure of the sporozoid, it is elongated, spindle shaped in nature. It has a nucleus, then different convoluted tubules, mitochondrion. The body is covered with a pellicle. There is a micropyle. Certain at the in the uh, apical region or the anterior part, you have the paired organelles, and the micro tubules are also present. These paired organelles and the microtubules, they are responsible for the penetration of this organism into the body of the host. Infection. So infection to a human being occurs in the form of sporozoites, and these sporozoites are actually present in the salivary glands of the female Anopheles mosquito. After the entry of the sporozoite into the blood during the blood meal by the female Anopheles mosquito, the asexual phase of the parasite begins. So the study of the different stages is very important. It is bit complicated. You have, what are the stages? You have the schizogony, gamogony, and sporogony. These three stages are completed by this parasite, either in the human being or in the mosquito. What is schizogony? Schizogony is simply the multiplication of the sporozoite that has entered into the human body after its entry into the either liver cells or the RBC, and to develop into large number of daughter cells di- through asexual multiplication. So this schizogony results in the increase in their number. Then what is gamogony? Gamogony, as the name implies, the formation of gametes. Gamogony means division of a stage where the gametes are produced, and these gametes are responsible for the sexual phase of the organism. So two types of gametes are produced, which we'll study, and the last is the gametes, they fuse to form the zygote, and from this zygote they undergo sporogony, that means production of sporozoite for reinfection or for infection to another animal or another human body. So we have schizogony, we have gamogony, we have sporogony. Remember, schizogony, it is a sexual division. Gamogony, it is a sexual phase, and sporogony, it is the asexual phase where the haploid sporozoite. Now, first thing is that when as soon as the parasite, when the mosquito bites a person, a, a mosquito laden with the sporozoite parasite, when it bites a human being, healthy human being, to obtain a blood meal, immediately the sporozoites, which are located in the salivary glands of the mosquito, they find a way into the bloodstream. Why? Because along with the secreted saliva, which contain anticoagulant not to allow the coagulation of the blood in its proboscis, these are freely released along with the saliva, which contains the anticoagulant, and immediately it gets cleared from the bloodstream and it heads to the liver. And in the liver, as soon as it enters into the hepatic cells of the liver, it undergoes schizogony, and this schizogony, or the asexual phase, it is completed through pre-erythrocytic schizogony and exoerythrocytic schizogony. Pre-erythrocytic, that means this schizogony stage does not occur within the RBC, it occurs within the liver cell. That is why it is called pre-erythrocytic. And exoerythrocytic, sometimes they continue the schizogony stage in the liver cell for the second time, for the third time. So that is called the exoerythrocytic schizogony. So in the pre-erythrocytic schizogony, it is also called merogony. The sporozoite, after entry into the liver cell, they change their structure into a spherical form or spherical state called the cryptozoite. Remember, it is called cryptozoite. This cryptozoite stage is called the trophozoite stage, and the parasite in this stage, they multiply. That means the nucleus breaks into nucleus divides into several times to form a multinucleate stage called the schizont. multinucleate scission. That means the nucleus are not completely separated. They are in a multinucleate state within the trophozoite stage called the scission. And this, the days, how many days it requires for the completion, it varies with the species of the malaria parasite. Now this scission, after attaining a certain stage of development, it ruptures, it breaks away along with the breaking of the liver cell. And this breaking of the liver cell, it liberates the uninucleate, these tiny nucleated cryptomerozoites. That means the daughter stage from the cryptozoite is called cryptomerozoite. Simply add merozoite to the crypto. So cryptomerozoites. What are cryptomerozoites? Merozoites are the daughter cells produced through from the cryptozoite through merogony or schizogony. And this schizogony is liver schizogony. So these cryptomerozoites, these are produced and they are released into the sinuses, into the cavity. And these merozoites, they have two options. They may enter into the RBC to start the erythrocytic schizogony, or they may reinfect the liver cell to continue exoerythrocytic schizogony. Remember, the number of times they continue a particular cycle, it is aimed to increase their number, but to a certain limit, because they know that if there is a greater load of this pathogen, it will result fatality with the host, and if the host die, they will also die. So that is why they continue depending upon the host condition, health condition. So they may enter into the RBC, or they may continue as in the liver cell, the schizogony phase in the liver cell, and it is called exoerythrocytic schizogony. Now, after exoerythrocytic schizogony, or this pre-erythrocytic schizogony, two types of cryptomerozoites, the cryptomerozoites which are produced, they are of two types. Uh, if it is a small one, it is called microcryptomerozoite, and if it is large one, it is called macrocryptomerozoites. Now, in general, it is believed that the resultant cryptomerozoites, which are not identical, they differ in their structure, they differ in their shape and size. The smaller ones, only they will enter the RBC, but the larger one have the potentiality to re-enter the liver cell and continue the liver schizogony as exoerythrocytic schizogony. So the deciding factor is the size of the cryptomerozoite. If it is micro, then it will continue. If it is macro, it will continue within the liver, and if it is micro, it will enter into the RBC. Then exoerythrocytic schizogony, if it continues within the liver cell again, that means they re-enter the liver cell. Each macrocryptomerozoite, remember I have told you that the larger ones will enter. Again, they pass through the same stage. They form a scission, and this scission is known as metacryptomerozoite. Sorry, these are called metacryptozoites, because in the first stage, in the first cycle of the liver, it is called cryptozoite. Again, if it is repeated, it is called metacryptozoite, because these metacryptomerozoites have resulted in the formation of metacryptozoites. They undergo schizogony in the same manner and produce larger number of daughter nucleus or daughter cells within the same metacryptozoites. Thousands of and these daughter cells are known as simply metacryptomerozoites. Metacryptomerozoites. The larger ones are called macro, and the smaller ones Micro, again, if the larger ones are there, they will enter into the liver cell, and the smaller one will enter into the um, RBC. Just a minute. Now, erythrocytic schizophreny, when either the pre-erythrocytic or exoerythrocytic cryptozoite. So, if the smaller form, either it is a cryptozoite or it is micro metacryptozoite, cryptomerozoites, whatever is it, if they gain access to the to the RBC, it starts the erythrocytic phase, and that is it is called as the erythrocytic schizophrenia. That means same type of multiplication within the RBC. So simply the merozoite, they get attached onto the surface of the RBC. They burrow within the into the RBC and assume a spherical shape, a rounded disc shape. They have a large single nucleus, and later on a non-contractile vacuole appears in the parasite, and the appearance of this vacuole pushes the nucleus of the parasite towards one side, and subsequent staining of the cytoplasm and the differential staining of the cytoplasm and the nucleus, it gives the appearance of a ring which you wear in your finger, and that is why it is called the signet ring stage. It is the most identifiable stage for the identification of the parasite in the blood film or the blood smear. This trophocyte, after entering, it grows at the expansion. It feeds on the hemoglobin of the RBC of the host, and this feeding of the hemoglobin, it gets decomposed into protein or simply amino acid and the hematin. The hematin, uh, these components of the hemoglobin, they are used by the parasite to synthesize its own protein. The ring configuration alters as the plasmodia begins to grow within the blood cell. So ring configuration, that means this ring stage, signet ring stage, it alters the plasmodia as the plasmodia begins to grow within the, it alters means it does not remain in this stage for a longer period. It shows, or the configuration changes. Full grown mature trophozoite. So what is trophozoite? It is the feeding stage. What it feeds? It feeds on the hemoglobin of the host RBC. Full grown mature trophozoite almost completely fills the enlarged corpuscle, that means red blood corpuscle, and now becomes a seasoned. Again, the seasoned. Okay, the seasoned. It becomes spherical. It becomes a seasoned. And the seasoned only will multiply asexually. First, the single nucleus will divide into many daughter nucleus by a process called erythrocytic schizogony. So, in the same manner that has occurred in the liver cell, same thing occurs in the erythrocyte or the RBC, and this is called erythrocytic schizogony or merogony. Now, sometimes the in the cytoplasm of these infected RBC, you find colored granules, and these colored granules, presence of these colored granules, these are known as the Schuffner's dot. Now, erythrocytic schizogony, the divides the by multiple fission. What is multiple fission? When the single nucleus divides into many fragments to form daughter nuclei, without taking into consideration of the number of chromosome. So chromosome does not here. So simply the many nuclei are produced, and many small uninucleate cells termed the erythrocytic merozoite. The earlier one was the liver merozoite. This is called erythrocytic merozoite, means the daughter cells that is produced through after erythrocytic schizophrenia. These daughter cells, they simply get arranged like the petals of a rose. So as you have seen the rose flower, the petals, they simply arrange like the petals of a rose, and that is why in this stage it is called a rosette stage. Particularly, rosette stage is most common in the Plasmodium vivax and falciparum. After some time, the cell membrane of the RBC, it ruptures, and these erythrocytic merozoites, they are released. And along with this release of this merozoite, a toxic product known as the hemozoin, they are also released into the free into the blood plasma, and it is circulated. The merozoites may continue the erythrocytic schizogony by reinfecting the RBC, and in each recurrence of the cycle, the parasite number gets amplified. Amplified means it just gets increased in its number. This hemozoin, it is a toxic substance, and it is believed that they are responsible for the rise in temperature of the host body. Then a rise in temperature. So this wave of fever will coincide whenever the erythrocytic schizogony is completed. That means jete thora RBC rupture habo, the erythrocytic merozoites will be released along with the hemozoin granules. Sete thora fever gudika recurrence habo. And after that, it will the temperature will rise up to 100, 203, and then suddenly, as the hemozoin granules will be cleared from the bloodstream, the temperature will certainly fall down until and unless the next stage of erythrocytic schizogony results in the liberation of the hemozoin. So this continue in a phase wise, and that implies the tertian and the quatern type of malaria, which deals with the recurrence of the fever in these species. So in each amplification of this parasite, RBC eventually is destroyed, and the large number of RBC, since it is destroyed, the patient becomes anemic. That means the less amount, the hemoglobin concentration decreases because the the RBC decreases. Fever occur every 48 to 72 hours, corresponding to the type of species that the person has been infected. Sometimes it does not follow the quatern or the tertian stage, because if a person is infected simultaneously with two or more species of Plasmodium, so each will continue. Or if the same species is infected within a gap of certain days, so that will continue. So remember, the occurrence of the fever coincides with the release of hemozoin granules into the blood of the host. Some Plasmodium vivax sporozoite do not immediately develop into exoerythrocytic phase merozoites, but instead produce hypnozoites. That means they become silent, they become dormant for periods of maybe several months to years together. Okay, they remain dormant, no symptom of malaria. And all of a sudden, after a period of dormancy, they get reactivate and produce merozoites. And that puzzles in some condition. when a when a person who has earlier shown the symptoms of malaria, again he says that he or she says that, no, I have not visited, I'm sleeping in the mosquito net, I have not visited mosquito endemic area, my clothes are always covered, I apply this insect repellent cream, so no bite of mosquito, but still I have the symptoms of malaria. That is due to the earlier hypnozoites, which are concealed and remain dormant in the human body. They are responsible for the recurrence. Then we after this schizogony, now it is gamogony. That means the formation of gametes. After repeated schizogony, that means multiplication phase, asexual multiplication phase, the parasite number increases, the load of the parasite increases in the host body, and they may either lead to acute anemia or death of the host, which the parasite never expects. The parasite will never wants that the host will die, because if the host is killed, if the host dies, then the parasite will also die. As a consequent, they cease the schizogonic stage and they go for the gamogony. What is gamogony? The formation of gametes. Why gametes are necessary? To continue the sexual phase. We have completed the asexual phase. Asexual phase occurs in the body of the human being, occurs in the body of the vertebrate host, that occurs in the liver cell and the RBC. So now in the sexual phase, it results in the formation of gametes. So in the earlier stage, we said that seasoned, it is gamont. Remember, gamogony, gamont, schizogony, seasoned. So gametes are responsible for the formation of gametocytes or gametes. Now, the stage prior to the gamete is called gametocyte, or the cell that will develop into the gamete, they are called gametocyte. So some of the merozoites in the blood cell, instead of continuing the schizogony, they simply increase in size and develop into sexual forms. Simply increase in size, no division, and they develop into sexual forms. Either they grow into the microgametocyte, which is small cell, or female macrogametocyte, which are larger cell. Female macrogametocytes, they are round and they are larger. Macro means they are very large, because always in this evolutionary history, all organisms, their female gametes are larger due to the they are laden with the food. So nucleus, due to the food laden or a deposition of the food, the nucleus is brought to one end or one side. It is eccentric in nature. On that basis, the position of the nucleus, we can easily we can easily uh, locate the presence of macrogametocyte and differentiate the macrogametocyte from the microgametocyte. The microgametocyte, they have a clear cytoplasm and they have a very central nucleus, because no food is required, and they give rise to the male gametes. Now these gametocytes, they develop as in this stage in the human being, but further development, they develop till gametocyte, but gamogony will never occur in the vertebrate host because of the temperature. They need a lower body temperature for the development of the gametes through gamogony. And you know, mosquito are cold-blooded animals. They have a lower body temperature, and when the female Anopheles mosquito, they bite, they give a bite for a meal, blood meal. At this stage of life cycle, the gametocytes are taken into the insect's stomach. They feed into onto the blood ray. The gametocytes, the RBC containing the gametocytes are present. They are taken along with the blood into their body. And in the body of the stomach of female Anopheles mosquito, this gametocyte will mature and give rise to the male and the female gamete. Now coming to the so gamogony is over. Okay, gamogony means the formation of gametocytes, and now the sexual reproduction. What is sexual reproduction? Just simply the fusion of the male and the female gamete. So the gametocytes are transferred to the body of the mosquito. They undergo the process of gamogony or gametogony, and these gametocytes, once they enter into the female Anopheles mosquito, they have resistance to the action of digestive juices and survive, uh, while all other accompanying stages get digested. Tautouchi, when the mosquito sucks the blood, in the blood, not only gametocyte stage is present, all other stages are present. That means the cryptomerozoites are also ingested, the merozoites are ingested. So in the body of the mosquito, except the gametocytes, all other stages, they are digested by the digestive juices liberated in the stomach of female Anopheles mosquito. Only the gametocytes, they sustain the action of the digestive juices. There is no nothing happens to these gametocytes inside the mosquito stomach. These gametocyte which are present in the RBC, I mean human RBC, the RBC rupture, and the gametocytes are set free. And these gametocytes, after they have become free, they develop into their respective gametes. Why I say respective gametes? Because macrogametocytes produce the female gamete and microgametocytes produce the male gamete or microgamete. In mosquito, the sexual cycle is completed within the 10 to 15 days, but it varies with different species of the parasite, and also depends upon the temperature. Gametes are developed through the process of gametogony. The microgametes, they are liberated from the microgametocyte. They are very active. You know, sperms are always active. The nucleus divides mitotically and into 6 to 8 haploid fragments. So they are mitotic in nature. Sporozoites, they are still in a haploid stage. So mitotic division results in the haploid fragments. That means the entire gametocyte develops into 6 to 8 fragments. That means 6 to 8 male gamete are produced, or 6 to 8 microgametes are produced. At the same time, the nucleus divides 6 to 8 fragments, and these nucleus, they they get collected in the periphery of the gametocyte. After that, the cytoplasm is extended, into which these nucleus are pulled, so that a flagellum like structure which are arranged along the rim of the gametocyte are found. So the cytoplasm push out from the surface of the gametocyte, into which each daughter nuclei passes, and ultimately exflagellation, by the process of exflagellation, the male gametes are released. And these, after their release, these flagellated uh, condition, or the spindle shaped, they are capable to move in order to meet the female gamete. Now, the formation of macrogamete, the macrogametocyte, they are responsible for the formation of only a single female gamete, which are ready for fertilization. After the formation, chemotaxis, through the process of chemotaxis, the female macrogamete, it releases certain chemical substances in the near vicinity, which attracts the microgamete to come nearer to the macrogamete inside the gut of the, or stomach of the mosquito, female Anopheles mosquito. A small conical structure is formed on the surface of the macrogamete, and this conical structure is called the reception zone. That means the region, it will not fuse anywhere. It will fuse in a particular region that is known as the reception zone. So a single microgamete is formed from each macrogametocyte. The fusion results in the formation of a zygote, and this zygote is a diploid zygote. This zygote becomes remains inactive, but after some time it becomes spindle shaped or vermiform or elongated. So when it becomes elongated or vermiform means worm like. When it gets elongated, it is capable of movement. What it does, it pierces through the epithelial lining of the mosquito stomach and simply enters, crosses the basement membrane, and becomes encysted there. It becomes a spherical oocyst or sporont. 50 to 500 oocysts are found in the stomach of a female mosquito. It is Sir Ronald Ross who found this stage of the mosquito, this stage in the stomach of the mosquito. So granular structures, they cover the entire surface of the mosquito stomach. And when he analyzed this granular structure, he found that the parasites are there. Then sporogony. So this diploid oocyst, it is in a oocyst means it is encysted. It is covered by a hard covering. So this encysted zygote is called oocyst, and it is present just beneath the basement membrane of the stomach wall. Again, now it is in a diploid stage. It enters asexual multiplication known as sporogony. So this sporogony leads to the formation of 1000 sporozoids, and after some time the wall of the oocyst burst due to much pressure, and the sporozoids are set free in the body cavity of the mosquito. So after the sporozoids, they are produced from a single oocyst. After the maturation of the sporozoid, the pressure causes the oocyst to rupture, and the sporozoids are released into the uh, nearby area. These sporozoids, they are capable to enter the mosquito, to invade the entire mosquito. That means they have a random erratic movement, and many of them, they find their way into the salivary glands, which is a favorable position where they position themselves waiting for infection to a new host when the mosquito feeds on its blood. At this stage, the mosquito is said to be infective. So when it bites healthy person, thousands of sporozoid. So a single mosquito can harbor 2 lakh sporozoid, and you can imagine a single bite can release around 1000 sporozoids in man. So this is a diagrammatic representation both in the human being and the sexual cycle in the mosquito. So it is a self-explanatory. It is divided into sexual cycle which occurs in the mosquito. The sporozoids are liberated. Sporozoids, you can see here, the sporozoids are liberated along with the saliva. It passes through pre-erythrocytic schizogony, that means in the liver, and again the it produces cryptozoids. Again, it may continue in the new liver cell, or the ultimately it produces merozoids. These merozoids, they enter the rbc they enter the rbc and complete the erythrocytic schizogony to produce a stage called the uh, micro to continue the merozoid stage. Each cycle results in the um, rupture of many rbc, destruction of many rbc, and in each cycle, with the destruction of rbc and release of these hemogen granules, the fever occurs. But sometimes some rbc where the trophozoites are present, they are not they will not develop into the second stage, rather they develop into the gametocyte. You can see here these gametocytes, they cannot develop in the human being, rather they wait to enter into the uh, cold-blooded animal, that is female, that is an insect, or the anopheles, where they give rise to the two types of gamete, micro and macro. They fuse together to form the zygote. Zygote develops into the ookinete. Ookinete develops into oocyst, and through sporogony, which in this sporogony stage, since it is a diploid, it undergoes meiotic division followed by mitotic division, so that the resultant sporozoite is a haploid one. Next, yes, this is a oocyst. You can see here, this is the stomach of the mosquito. These are the, since it is a insect, so there are malphigian tubules. These are concerned with the excretion. Okay, the nitrogenous excretion is through malphigian tubules. And the same thing, the same function as the human kidney does. And this is the intestine. You can find that these oocysts, they protrude, they appear like granules on the surface of the mosquito stomach. This observation has been observed by Sir Ronald Ross, which confirmed that the mosquito are the carriers of this parasite. So here a small diagram where you can see inside the mosquito stomach how this exflagellation occurs. You can see here the exflagellation occurs. This is the stage. This is the zygote stage. Sorry, this is the gametocyte. This gametocyte may give rise to the macrogametes, or it may give rise to the microgamete. So one microgamete, that is the male gamete, and one macrogamete, they fuse together, and after fusion the zygote is formed. The zygote become vermiform or worm like, so that it can pierce the wall of the stomach of mosquito and then get covered by a cyst covering known as the oocyst. And in this oocyst stage, the nucleus, the diploid nucleus, undergoes meiosis followed by mitosis to form large number of sporozoites. And when the sporozoites have built up in a large number, it bursts the wall of the uh, oocyst, and these are released. So after release, they wander about the body, and some find their way into the salivary gland of the mosquito. And after salivary, when they reach the salivary gland, when the mosquito bites, along with the anticoagulant present in the saliva, they are released into the blood, and the cycle follows. So these are the hepatic cell, liver cell, again continuation of the liver cell. After several cycle, they now go into the RBC, and inside the RBC they again multiply, but after few multiplication, they give rise to the gametocyte, and this gametocyte will enter into the body of the mosquito for the formation of the gamete. Now here you can see, this is, see here, this one, this is the, all these are all RBC. Okay, RBC, human RBC, they are non-nucleated, no nucleus. But you can see here two stages, two signet ring stage. You can see here the dot like structure and a vacuole. Due to the appearance of the vacuole, it is called the signet ring stage. So B, these are the trophozoite stage, and these are the seasons that are formed, okay, during the schizogony process inside the RBC. Okay, now so do you have any doubt in the life cycle? We have completed the life cycle. Mm, if you have any doubt, it is clearly
No, sir.
shown.
No doubt.
Okay, just a minute.
Sir, which type of question come for this chapter? Is the life cycle of
Yes,
Plasmodium?
life cycle, or the asexual cycle, the life cycle in the human being, life cycle in the mosquito, about the pathogenicity it causes. These are the questions. Okay. Thing is that the parasite tends to increase their number, increase their virulence, increase their number in the human body, and it is the best example that is exhibited as alternation of generation, where the sexual phase alternates with the asexual phase. So sometimes the question may say, what is alternation of generation? Uh, Mention its significance relating with the life cycle of Plasmodium. Okay, thought-provoking question. So significance means the this due to this the two stages, they have the chance to multiply. They have the chance to increase their number, their load inside the host body. Because all organism, there is a tendency to increase their number. There is a tendency to increase their uh, number.
Sir, I have a question.
Please.
Sir, ring stage of RBC is seen in which plasmodium?
Just a minute, just a minute. Uh, huh. Please carry on.
ring stage of RBC is seen
Yes.
in which plasmodium?
Which plasmodium?
Yes, yes.
Ring stage is observed in all, particularly in vivax and falciparum. Okay, the
Okay,
signet
okay.
ring stage. It is the
The
trophozoite
lab
stage.
the laboratory diagnosis is important for exam.
Laboratory diagnosis means initially when the antigenic testing was not, this card testing was not available. Initially, in our days, they simply take a film of the blood film of the person suffering in order to,
Smear, thin smear.
yes, to go through the presence of signet ring stage, because it is only this stage which can differentiate from other cells.
Okay, okay.
Now, regarding pathogenicity, pathogenicity means what are the effect the parasite has on the human body. That is called the pathogenicity. And here, that is febrile paroxysm. That is the attack, the cyclic attack of the fever, along with the um, shivering. Okay. So cold stage, hot stage, sweating stage. So cold stage, it remains when you just shiver. That is the cold stage. It is this stage initiates with lethargy, headache, nausea. All these are, and chills, or kompana hobo, chilling, followed by rigors, just shivering of the body. Then again, it occurs for 20 minutes, followed by hot stage. The temperature suddenly rises, and this temperature rise, it may rise up to 102, 3 or 4 degree uh, Fahrenheit. Patient feels burning hot, and at that time, if the temperature goes beyond 105, it is necessary to wipe the entire patient's body with wet cloth. ସେତେବେଳେ ତାକୁ sponge bathing କରାଯାଏ, and that too, care should be taken, the uh, 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 in the forehead, the pani patti pakai deba katha, because forehead is the region where maximum circulation, and when this blood gets cold, so remaining part of the body also get cold. So that is why ଆମେ ମୁଣ୍ଡରେ ଯୋଉ ପଟି ଓଦା ପଟି ରଖୁଛେ, ସେ that is the reason, because cerebral circulation ସବୁଠାରୁ ଅଧିକ ରହିଥାଏ in the brain. And there is a sweating stage ଦେଖିବ, ତାପରେ ଝାଳ ବୋହିବ, and shivering stage ରେ ତମେ ଘୋଡେଇ ହେଇ ପରିବ କମ୍ବଳ ଫମ୍ବଳ ସବୁ, ତାପରେ sweating ହବ, and you get rid of it. So this is the febrile paroxysm, and the duration of the fever and the recurrence of the fever, it depends upon, the species the person is infected. Accordingly, the tertian fever, 48-hour cycle, where it is occurs in vivax, falciparum, oval. Cotton fever only in malaria, Plasmodium malaria, every 72 hours. It has a 72 hours cycle. And quotidian, every second day. So every second day means when a person is infected simultaneously with multiple species of plasmodium, so there is a chance that every second day or more frequently they will come with the fever. So in such case, you cannot simply say that no, it is not a malarial fever, because malarial fever will occur every third day or every 2 days or every 4 days, because only it will reveal with the test, by blood test or something. Now, another condition is that anemia, when the blood is destroyed, RBC is destroyed, so hemoglobin is destroyed. There is less amount of hemoglobin, and the condition that is when less amount of hemoglobin, ତାକୁ ରକ୍ତହୀନତା କୁହାଯାଏ, and it is called the anemic patient. So sometimes the hemoglobin level, generally it goes below 9 gram per dl, deciliter. Okay, hemoglobin is measured in gram per deciliter. Deciliter means every 100 milliliter of blood, usual concentration is 14 to 16, depending upon the sex. But in such cases, it may go below 9, and sometimes the fatality results in when it drops below 3 gram per deciliter. and in such state, the person dies. The mortality rate increases with the decrease in the hemoglobin concentration of the blood. Then another condition is splenomegaly. Megaly means enlargement. মানে স্প্লিনটা বড়ি যায়, so spleen ro weight body যায়, তার size body যায়, and it acquires grayish black coloration. There is a congestion and a reticuloendothelial hyperplasia. Now reticuloendothelial system will come across. It is this system which deals with the macrophages, which are responsible to kill the worn out cells along with the RBC which has completed 120 days. So it RBC गुड़िक सब ये जाउछ. It so splenomegaly means enlargement of the spleen. Then pernicious malaria. It is a grave form of malaria. It is caused by falciparum. So it is occurs due to blockage of capillaries because of the parasitized RBC. So when the capillaries get blocked, and this blocking of the capillaries by RBC is called agglutination in the capillaries. Agglutination means just they get assembled. So assembly agglutination is directly related to the appearance of sticky nature of the RBC. They get agglutinated, they get collected in a particular region, thereby blocking the pathway of the capillaries. And if these capillaries become, it is blocked in the cerebral region, you know what will happen, because that particular area will not get blood and necrotic activities will occur, thereby leading to the uh, paralysis of certain regions. So this is pernicious or continuity malaria, and this malaria, it may be cerebral malaria, as you know, caused by this falciparum, or septicemic malaria, characterized by high and continuous fever, pneumonia, sudden drop in blood pressure, and reduced blood flow to the brain, and other, that is called septicemic malaria, which ultimately leads to fatality. Algid malaria is the malaria characterized by cold and sticky skin and vascular collapse, accompanied with vomiting, watery diarrhea, dysentery, and bloody stool. So sticky skin means sweaty skin. The skin always it will sweat. There is the enlarged liver also. Liver is enlarged because liver also contain reticuloendothelial cells, and the proliferation of these reticuloendothelial cells in the liver as well as spleen are responsible for the enlargement. That is called hyperplasia of the spleen and liver. So in case of spleen, it is called splenomegaly. In case of brain also, cerebral edema. Edema means enlargement of the brain may occur, or congestion may occur. So if the capillary of the blood vessel, capillary blood vessel of the brain, it is clogged with parasitized RBC. Why we say what is parasitized RBC? RBC which has been infected by the parasite. RBC which host the parasite. That is called parasitase RBC. So if it is clogged, because it is believed that they develop a stickiness nature, so it causes the clogging of the RBC and nervous. If the clogging occurs in a particular, that is the degeneration of nervous tissue leading to the appearance or parasite paralysis of certain region of the body. So kidney
Sir,
also,
how, sir, may I ask question?
yes.
Sir, how the sickle cell disease register malaria?
Yes, it's a good question. Now, sickle cell anemia are of two types. Okay, um, in persons with both the gene, when persons who have normal blood cell as well as sickle separate blood cell, they will survive. But the person who have all the blood cells are sickle shaped, they will not survive beyond the age of 16, because beyond the age of 16 or in adulthood condition, because they cannot sustain provide the energy uh, demand by the body. So energy demand by the body is directly related to the oxidation of the food, and oxidation of the food is directly it is met by the amount of oxygen. Now the blood cells, because they are sickle shape means ardha chandra, moon shaped structure, it will have less amount of RBC, hemoglobin. So the oxygen carrying capacity decreases. Now they have a resistance for to the malarial parasite, because malaria parasite, they increase in their size, they become a trophozoid, they become enlarged, they become amoeboid. But the sickle cell will not provide space for their development. That is why the sickle cell patients, they are saved from this disease. They don't acquire with disease. Am I clear?
Yes, sir.
Okay. Now heart also, may become dilated and taku swollen heart ame kahiba, dilated and flabby in nature. Lungs, they become congested, edematous means fluid filled. So it may lead to the pneumonia or respiratory distress syndrome. Anything that interferes with the functioning of the lungs ultimately leads to pneumonia of different types. Then in the gastrointestinal tract, it also becomes edematous, congested with small vessels of the intestinal mucosa. Then pregnancy loss, sometimes the if the mother is suffering from malaria, this and the malaria is severe, then the mother suffers from anemia, and this anemic condition is very dangerous to the growth of the fetus. Now, relapse of malaria, as I told you, hypnozoites are responsible for the relapse. আমি କହିବା ଯେ ବହୁତ ଦିନ ହେଲା malaria ହେଇନି, ମୁଁ ମଶାରୀ ତଳେ ରହୁଛି, this thing that thing, but suddenly malaria ହେଇଗଲା. And the reason is that they found that certain hypnozoites ସେଟା ଲୁଚିକି ରହିଥିଲେ, dormant ହେଇକି ରହିଥିଲେ, active ନଥିଲେ, and suddenly they have become active. That is called the recurrence or relapse of the malaria. Now, malaria detection, nowadays it is very easy. Earlier days, there are two types of smears, thin smear, thick smear. It depends upon the thickness of the blood film. And in the thick smear, it can detect even low levels of blood, because the volume of blood adhika rahila, then low level of parasite rahile matya, since the volume blood adhika rahila, there is chance of detecting the parasite is greatly increased. But in thin blood smear, as I've told in my slide, it is very clear. Each RBC ame dekhi pariba, and we can view whether it contains, after proper staining, we can stain the nucleus of the signet ring stage, which stain with a darker color than that of the cytoplasm. Now, antigen jo dipstick roichi, antigen detection re ame both we can identify or detect the presence of this malaria within no time. That is called a dipstick test. Then molecular diagnosis, also the PCR, the polymerase chain reaction to find um, the presence of the nucleic acid of malaria parasite when it is multiplied, when it is um, polymerase chain, that means its quantity is increased, and through proper way we can justify the presence of these nucleic acid of the malaria parasite. Then serological test, you can the presence of the antibody in the patient. So blood plasma ko we can subject it to ELISA, that means enzyme linked immunosorbent assay test re ame jani pariba, we can easily come to understand whether the antibody of the malaria parasite is present or not. Now, the immunity and immunity evasion, natural immunity amar to sadabela roichi. So natural immunity, the human being has a natural immunity to defend against any type of the infection or any type of foreign material which try to enter into our body. But the individuals who reside in malaria endemic area have suffered from the infection, or they develop some degree of immunity, natural immunity. Okay, so simply with few bites they don't develop malaria, or they develop a very weak malaria. So this natural immune responses, it lowers the risk of the disease to the person who have earlier exposed to malaria. Then adaptive immune system, it is the second line of defense. It is mediated by the T cells and B cells and the antibodies, which we have already studied yesterday. So in the human being, there are various types of these adaptive responses mediated by B cells and T cells that are responsible for the formation, production of antibody against this malarial parasite. Um, then anti-malaria immunity, it confers protection, but till now the complete immunity has not been achieved. Then we can the immune system also can act against the sporozoites. So when the sporozoites are inoculated through the skin, it affects the place for uh, antigen presentation and the location and type of response. So neutralizing anti sporozoite antibodies. So neutralizing anti-sporozoite antibodies can block the. So this is the evasion technique showed by the parasite to the host immunity. ता माने होची parasite मध्य किछि अस्त्र prepare କରି ରଖିଛନ୍ତି, so that they will not allow the host immune response to kill them, to remove them from their host body. So one such is that against a sporozoite, if the host immune system is against a sporozoite, the sporozoite have developed the capacity to neutralize the antibody that develop in the host body. Then they block the motility of the neutralizing anti-sporozoite antibodies, can block the motility of sporozoite, prevent invasion of sporozoite in the hepatocytes by preventing the sporozoite ligand to interact with the hepatocyte receptors. So then against hepatic stages, during hepatic stage cellular response, here also different types of cytokines and immunogenic responses come to play. These include the CD8 plus cells, then interferon Y gamma. T cells kill the parasite by the release of cytokines or by a different method, or activating the macrophages, they kill them. And against erythrocytic stages, so hepatic stages, sporozoite is the initial stage. We can block the motility of the sporozoite by different means, prevent the sporozoite entering the. If the sporozoite have entered the liver cell, we the T cells can employ to kill this parasite through the action of cytokines. If they have entered the RBC, immune response during the erythrocytic stage is mainly mediated by antibodies. When they have entered the RBC, the antibodies come into play and they make these parasites or the antigens effectiveless. Now, immunity evasion technique by parasite, they have shown how to avoid the immune response of the host body. So antigenic polymorphism, that means they show different types of antigenicity. That means a fixed type of antigen are not present. So if we prepare a particular vaccine or a particular molecule against a antigen or antibody against antigen, the next time within years the antigen will change, so they will not show any response to this vaccine. So that is called antigenic polymorphism. Polymorphism means many forms of these antigens. So plasmodium, which is a haploid organism, is prone to mutation. Antigenic polymorphism means if they change their antigen What are antigens? Antigens are glycoprotein molecules or glycolipid or protein molecules present on the surface of the cell, by which a particular specific cell can be identified as a foreign cell or a own cell. All types of protein are controlled by a particular gene. So if a particular gene controls the formation of a particular antigen, suppose antigen X, if this gene which produces the antigen X becomes mutated, becomes changed, it will lead to the formation of a different type of X, a different type of antigen. It may be X1 or it may be X2. So under such condition, it is not possible to target the antibody. So they fool our immune system by forming many type of antigenic antigens. Then antibody dependent enhancement of infection. So antibodies against these CSP enhance sporozoite entry and development in hepatocyte. Those against the uh, falciparum asparagine rich protein increase merozoite invasion of RBC, um, while merozoite specific antibodies in conjunction with complement can also facilitate RBC invasion. So merozoite specific antibody in conjunction with complement can also facilitate. Say they just mask जोठी जो molecules गुड़िक produce होची ताकू avoid करिबा पई ताकू न चिनिबा पई they for different methods, that is called the evasion mechanism. Antigenic diversion. Antigenic diversion means when the action of inhibitory antiparasitic antibody is prevented by non-inhibitory antibodies. So antigenic diversion means antibody produce hegala, it will start acting against the specific pathogen, but simply it is prevented by non-inhibitory antibodies. So neutralizing antibody which block the cleavage in the C-terminal part of the antibodies can prevent invasion. So, however, antibodies against the adjacent or overlapping region can block the effect of these antibodies, allowing invasion of merozoites. So, whatever structure that does not allow the entry of the, if it is evaded, if it is blocked, the entry of the sporozoite or merozoite into the respective cell can easily take place. Epitope is the particular region of the antibody that understands the antigen. So if the epitope is masked, if the epitope is covered, it cannot recognize the antigen, and the pathogen or the specific parasite will gain entry into our body. So the vaccine strategy means nowadays what are the types of available vaccines, and whether the vaccines are effective or not, or whether they are in a trial mode. So there are different types of vaccine that has been designed for this protozoan parasite. It may be R21 vaccine. So the naming is done by the concerned pharmaceutical company, which involves the different types of gene. So R21 vaccine is composed of a single subunit. It is proposed that higher proportion of CSP and less amount of may lead to improved immunoglobulin G responses in comparison to. So R21 vaccine, then CSP based RTS vaccine, these are under trials. Now promising targets for vaccine means if a pharmaceutical company they want to prepare a vaccine, they will first decide which thing we will target. ମାନେ କୋଉଟାକୁ ସେ ପାଥୋଜେନର କୋଉ ଜିନିଷଟାକୁ ଆମେ ଟାର୍ଗେଟ କରିବା, ସୋ ଦ୍ୟାଟ ସେଇ ଆକ୍ଟିଭିଟିଟାକୁ ଆମେ ଇଣ୍ଟରଫିୟର କଲେ the pathogen will die or it will become functionless. That is called the promising targets for vaccine. For example, in the ପ୍ରଥମରେ thrombospondin related adhesive protein is a parasite antigen. It is transmembrane protein containing adhesive domains. So it's a parasite antigen. ଏଥିରେ adhesive domains ରହିଛି which brings about stickiness, enable the motility of the sporozoite in mosquito and humans, mediating their migration from the skin to the liver. যদি এই প্রোটিনটাକୁ ଆମେ ଟାର୍ଗେଟ କରିବା, it is considered a major target for the development of malaria vaccine. এই প্রোটিনটাକୁ ଟାର୍ଗେଟ କରିବା, if this protein is not synthesized, that means this motility of the sporozoite in mosquito and human will cease. Now infection hobani, so that is called the target. Now prophylaxis means kon kariba, so how to protect yourself. So first thing is the community level re and individual level re, um, from the individual level re, from larva, against the larval stages and against the adult stages. Larval stages means prevent water, so mosquito develop from the larva. So if you don't larva dwells in the water, if you don't allow water to remain stagnant, the larva will not develop, the mosquito will also not complete their life cycle. So adult stage, simply the adult mosquito prevent the approach of the mosquito to come near the vicinity of your body. So screening of the doors, windows, applying intoxic these chemical coated um, mosquito net, then spraying of insecticide in the nearby bushy area, keeping the overhead tanks covered, and those items which are not required, or which are there is a possibility that they will allow water to become stagnant, it should be simply covered or simply thrown away. It should not be kept in the nearby area. So treatment, there are different mode of treatment. There is a quinine, chloroquine. These are the medicine that are given. Mefloquine, primaquine, artemisinin. All these are different drugs that are aimed at in different stages of the parasitic life cycle. Now,
Sir, I have a doubt.
please go on.
Black water fever is caused by which
Black,
plasmodium? Black
black,
water fever.
black
Water
water,
fever.
black water.
Yes, yes. Black
It is
water fever.
caused by
Which plasmodium?
black water fever. Where did you get? Hmm? Black water? नाटा काहीं केमती जानलो?
ଗୋଟେ କୋଶ୍ଚିନ୍ ଦେଖିଥିଲି ତ ସେଥିରେ
हाँ,
ଆସିଥିଲା।
नहीं फाल्सीपारम रे होची। So
ଆଜ୍ଞା।
काहीं की कुआ गला जो ब्लैक वाटर फीवर?
ତ ସିମ୍ପଟମ୍ ଟିକେ ଅଧିକ ଡେଞ୍ଜରସ୍ ନା କଣ ସେଟା ଶୁଣିଥିଲି କଣ ଶୁଣିଥିଲି।
जनरली फाल्सीपारम तो बहुत डेंजरस रही। सो एथिरे
ହୁଁ।
रोची कोन दी आरबीसी, तो निश्चित भाव आरबीसी गुड़ा को बंद ये जो भांगी जाउची, रप्चर हे जाउची, आउ रप्चर हे गला माने जो मेरोगोनी सिजोगोनी रे हेमोग्लोबिन रिलीज हे जाउची. So sometimes these hemozoin granules in large concentration, they may become present in the urine. तो से यूरिन रे रहिबा जोगु
ସେଥିରେ ଗ୍ରେନ୍ ମ୍ୟାଲେରିଆ ବି ତ ହେଇପାରୁଛି ନା ଫାଲ୍ସିପାରମରେ।
हाँ नहीं सेटा होची
ଆଜ୍ଞା।
कोन की को स्टेट रे रहिची, जदि अत्यधिक हे गला ना, ते जेते
ଆଜ୍ଞା।
अधिक आरबीसी डिस्ट्रॉय होची, आरबीसी रो हेमोग्लोबिन जो हेमोसाइन ग्रैन्युल्स गुडी को दे बिकम लॉस्ट, किंतु जेते प्रोडक्शन अधिक होची, समटाइम्स दे अपीयर इन द यूरिन ऑफ द पेशेंट, एंड दिस यूरिन रे जो हेमोसाइन और द metabolic byproduct of the hemoglobin जो बाहरिबो, it renders dark color to this urine, and that is why it is called dark water. ସେଥିପାଇଁ କୁହାଯାଉଛି। So treatment ଆମର ସରିଲା। So prophylaxis ame jemti how to keep ourselves from the bite of the mosquito. So au kichi doubt rahila ki students, or we'll move on to our next topic, that is leishmania.
Nice, sir.
Okay. So is the slide shared? So leishmania, it is also a blood parasite, leishmaniasis. So this leishmania morphology, we can see here. Now, leishmania species, there are basically, it is a intracellular, okay? Intracellular, like that of the malaria parasite. It is a digenetic flagellate. So mosquito, this plasmodium is not a flagellate parasite. It is a digenetic flagellate parasite. That means it completes its life cycle in two hosts. And these two hosts, one of them is simply the human being, and the other is the sandfly. Okay, sandfly. So three types of this leishmaniasis are found. Three types are found. Visceral. When this leishmania in these two forms, pro-mastigote form and a-mastigote form, are present, when these are present in the body, particularly in the internal organs, viscera mane hochi internal organs. So when they're present within the body in the internal organ, intracellularly, particularly the reticuloendothelial system, it is called visceral leishmaniasis. Cutaneous mane semane they will show their presence in the skin. So they may form certain sores. So it is caused by Leishmania tropica, and the visceral is caused by Leishmania donovani, and they cause local lesions. Lesions mane tika ulceration type of skin ulceration dekha jibo, ta cause local lesions. Local lesions means at the region where the sunfly has bitten. Then mucocutaneous mane mucus covering skin, where the so mucus covering skin hochi basically our mouth cavity is mucus covering skin roichi, nasal cavity mucus covering, particularly in the areas of skin and oronasal mucus. Oronasal mucosa. So nasal mucosa as well as oral means pharyngeal or mouth cavity mucosa, it becomes inflamed due to the presence of this Leishmania mexicana, and it causes local skin lesions. Localized skin lesions means ulceration which is localized. Always a primary host is a vertebrate host, and the secondary host is the sandfly, which is an insect, and the vector for this disease leishmaniasis, they are the vectors. This it occurs in two stages. One is called the pro amastigote and the pro-mastigote form. A mastigote form, it appears, you can see here, it is a globular in shape, oval in shape, a mastigote form. No flagellum is present, and it is the intracellular stage. It is present in the human being. So when it is present in the intracellular stage, it cannot move. So there is no locomotion. So no flagellum is required. So seta hochi a mastigote form or leishmanial form. It is present in the human host. It is intracellular or within the cell, and particularly they are found in the reticuloendothelial system of the human being, which includes the spleen, which includes the liver and the lymph organs. So reticuloendothelial system, as I've told you, they are the cells concerned uh, with the protection of the body, with the formation of plasma cell, with the formation of macrophages. So they, if these parasite, they infect these regions, that is spleen, liver, and so on. So a mastigote form are shown, or they are present in these regions, that is called the reticuloendothelial system of the infected human. Now coming to the promastigate form, which is the flagellated form, and this flagellated form are only present in the sandfly. তাহলে আমি কোন জানিবা human being রে जो state রয়েছে without a flagellum, because it is intracellular, it is not required to move. And in the sandfly it is with a flagellate, because it requires to move. So early stages of this promastigote form, they are short pear shaped or oval forms, because they enter from the human body into the body of the sandfly in a state where there is no flagellum. That is called the early stage of promastigote. They are pear-shaped oval forms, and subsequently, after their development, they become spindle-shaped, they become slender, they increase in length, the development of the kinetoplast and the formation of the flagellum from this kinetoplast, they occur. After these develop, they are now capable to move in throughout the body. Uh, just a minute. Yes. So here you can see the life cycle. First, let's study the life cycle. So in this, how the parasite, it changes between the human host and the sandfly vector. So there are, it is numbered. Now, number one, sandfly, they in the within the sandfly, the parasite is in the stage of promastigote, which is a flagellated in the flagellated form. Okay, flagellated form. And the sandfly, they are scientifically known as Phlebobotomus argentypes. Phlebobotomus argentypes. Sandfly. The promastigote form, which is the flagellated form, they are ingested by the immune cells, macrophages, by the process of phagocytosis, where they develop into a mastigote. So when it is infected, when the human being is bitten by the infected female sandfly, remember, female sandfly, female mosquito, female Anopheles mosquito, female sandfly, they carry them in their salivary glands. So when it enters into the body of the human being, immediately these are taken up. The promastigotes are ingested. These are taken up.
Screen is not visible.
Screen is not visible.
No, sir.
Uh, I don't know how can I, because my son is he has gone outside, so I cannot. So you just do something in your, you can enlarge it. Okay, otherwise I
Okay,
may
sir.
lose this screen. Okay, and let me see if I can.
No problem, sir. You can say.
Okay, yes. Is it now? Okay, I have increased it. Is it okay now?
Okay, sir.
Huh.
It is not visible, sir. Not visible.
Not visible. Not visible.
No, sir. Sir.
Now is it
Only
visible?
books are visible, sir. Only your
There is no any
only we
problem
can
in the
able to
class.
see your books, sir.
Oh, oh, acha, acha, acha. Then,
You are clearly visible, sir. I think there is some network problem in that area.
now I have closed the sharing. Now, what is the condition now? Tell me.
No, sir, you are clearly visible. There is no any problem.
I am visible, but what about this?
The screen is not visible.
Okay, let me see. Just a minute.
Let's proceed, sir. Let's go, sir.
Okay, okay. Just a minute.
Your voice is also clearly audible, sir. Please proceed.
Sir, please continue.
Sir, we can able
Sir, it's visible now, sir.
now completely visible, sir.
Okay.
Now we can able to see it, sir.
Okay. Thank you. Thank you. Thank you. Thank you. Now, the life cycle in this stage, the flagellated stage, it is present in the salivary glands when the insect has bitten the human being. Immediately, these pro-masticate stage, they are taken up by the by the immune cells, particularly the macrophages, by the process of phagocytosis. So they are ingested, and here they are not killed, rather they develop into a masticate form. That means they lose their um, flagella and they change into their structure. The amasticate form of the parasite, they multiply and divide inside the macrophages. Simply, all parasite, human blood parasite, they simply increase their number by dividing. So simply they divide inside the macrophages, which is present in the reticuloendothelial system by binary fission. So binary fissions means from one organism. It is not multiple like that of the malarial parasite. It is a binary fission. Only two products or two daughter cells are produced. The multiplication will continue till the cells are packed with the parasites. So from one promastigate, two daughter cells, then from two, four cells. It will continue unless and until the parasite number becomes 50 to 200 within these macrophages. And subsequently, the macrophages will rupture. They enter the, after rupture, they enter the circulatory system of the human being and infect new cells of the reticuloendothelial system. Means they again continue in the macrophages. जेमती आरबीसी protozoan parasite, I mean, the malaria parasite continue kartala. Again, here they will reinfect new cells of the RES. RES means reticuloendothelial system cells, the macrophages. Successive infection of new cells and increase in the total number of parasite inside the host body rapidly increases the destruction of the RES cells. So, what is the consequence? You know, the consequence is that the reticuloendothelial cells, when it is repeatedly infected by this parasite, there they become destroyed. And during the circulation of the amastigate form in the human blood, a few parasites are phagocytosed by the neutrophils and monocytes. Now, in the life cycle, number two, it is taken up by a macrophages. You know, it looks like an amoeba. So macrophages, they have multiple extension of finger-like structure so that they can engulf. After engulfing, the promastigate forms, which is the flagellar stage, it is converted, transformed into amastigate stages inside the macrophages, which is a cell of the reticuloendothelial system responsible for the protection of the body. In the next stage, the A mastigotes, they again they enter the fresh macrophages, again multiply. So multiplication is through binary fission. And after the load, then ultimately the sandflies, when it bites the human being, they ingest, along with the macrophages, they ingest the infected macrophages, which contains the A mastigotes. The A mastigotes, after entering into the sandfly body or sandfly gut, they get transformed into the promastigote form in their gut, where after attaining or after forming the kinetoplast, after forming the flagella, they again increase in their number by uh, through they multiply through binary fission, and that is longitudinal binary fission. And then they migrate to the proboscis or to the salivary glands, where they wait for the transmission to the next healthy human being during their bite, or when they bite the human being. Now, sandfly biology in the leishmania life cycle. So, vector in case of uh, malaria parasite, the vector is female Anopheles. In case of leishmania, the vector is sandfly. Now, this sandfly, they have some type of reaction towards the leishmania. So, it is the life cycle is confined to the midgut. Okay, midgut, the intestine or middle part. So, in sandfly, the life cycle of leishmania, this parasite, protozoan parasite, it is only confined to the midgut. The morphological transformation means the development of the flagella, the elongation or of the parasite. It occurs in the midgut of the sandfly. Now, it is triggered. Why it develops? Why not in the human being? Because same formula, it requires a lower body temperature, and this lower body temperature is provided by the sandfly, which is a cold-blooded animal like all other insects. So, human being, if these are not taken by the sandfly, they will not develop, because their further development into a mastigate form requires the lower body temperature. That is it. Now, Leishmania development. What are the natural barriers? Natural barriers to Leishmania development. So, within the sandfly gut or within the alimentary canal of the sandfly, there are natural barriers. Barriers means the factors which will not allow the development of the Leishmania. These include proteolytic enzymes. So, proteolytic enzymes are the digestive enzymes in the sunfly, is one of the critical factors affecting Leishmania development in the vector, and represents attractive targets for vector-based transmission blocking strategy. Vector-based transmission blocking strategy তা অর্থ কোন? Now, when we come to know that the midgut releases certain proteolytic digestive enzymes which will not allow the activity of the leishmania, so we can target such a type of enzyme to prepare our drug. So vector based transmission blocking strategy, these enzymes such as trypsin, chymotrypsin, it reduce numbers of parasite in the midgut and can even destroy them. So this strategy, this characteristic feature, we can employ for the preparation of our drug against the sandfly. Consequently, leishmania has developed mechanism. So, if this is true that the um, barriers are present, proteolytic digestive enzyme barriers are present, but still they don't act against the leishmania. That means the leishmania have developed certain mechanism to resist the proteolytic attack of without inhibiting or affecting the overall. So this action by which it can resist, we can prepare the drug which will not allow them to resist this act of the proteolytic enzyme. Now, peritrophic matrix surrounding. So, peritrophic matrix surrounding the ingested blood meal. So, when the blood meal is ingested by fly, immediately a covering that is called the peritrophic matrix. A covering is produced. The proteins involved in its formation, this covering, the surrounding uh, the maturation and the application provide a promising target for transmission blocking vaccines. So, ଯେତେବେଳେ ଆମେ ଜାଣିପାରିବା ଯେ this blood meal is covered by a peritrophic matrix surrounding the ingested blood meal, and it involves the formation of certain type of protein. So if we block the formation of such protein, no peritrophic matrix will be formed, and these it cannot further, these parasites will not continue their life cycle. So this PM is secreted by the midgut epithelium. So it is necessary for further development. So if we can resist or not allow the development of the PM, that is peritrophic matrix, we can kill, or we can halt, or we can block the progress of this disease in the community. Then sandfly immune reaction, innate immune response plays an important role in the control of bacterial and parasitic infection in sandfly midgut. So sandfly, they have an immune response to control the bacterial and parasitic infection. So they also release antibacterial peptides, defensins, which exhibit antiparasitic activity. So they are not killed. They don't have their natural enemy. So if they have a natural enemy, or we can target a natural enemy, then if we control the population of the sandfly, automatically we can control the spread of this leishmania disease. So other components of the innate immune response include the pattern recognition. Now this parasite, it develops in the midgut. So the parasite need to bind to the midgut epithelium too. So how they bind, they just get entrusted. পুরা মানে গোটা spiky form রে they get entry into the midgut epithelium, so that they will not be carried over along the gut, gut. So to avoid being excreted with the blood remnant. So যদি ব্লাড গুড়িক মুভমেন্ট করছি, ফুড মুভমেন্ট করছি, they become anchored to the midgut epithelium to avoid their removal from the body, from the sandfly body. So after their escape from the endoperitrophic space, the parasite insert there. How do they get anchored? They get anchored by inserting their flagella in between the epithelial microvilli. Microvilli are the projections from the anterior surface of the epithelium. So in between this microvilli, they render their flagella and get anchored. Now, role of sandfly in midgut microbiota, the digestive tract of sandfly, it colonizes various microorganisms. ସବୁ animal, ସବୁ organism ର microorganism ରହିଛି digestive tract. Human being ରେ ମଧ୍ୟ biota ରହିଛି. So when we take antibiotics, so we are instructed to take probiotics along with the antibiotics, because antibiotics, they kill the important microflora of our digestive system, that will subsequently lead to the improper digestion of the food. That is why we are asked to take probiotics. So similarly, sandfly, this midgut, they have certain microorganisms which they have acquired from the soil, the food in which they breed at the larval stage, or through contaminated sugar mills at adult stage derived from plant leaves, fruits, or aphid. So another source of microorganism for sandfly is the skin of sandfly host obtained while ingesting blood meals. The sandfly associated symbiotic, they are symbiotic. Okay, they play a symbiotic role. So symbiotic, that means there is a close association between the host, host hochi, sandfly, and the parasite, the member. Okay, the association between the sandfly and the microbiota in their midgut. So it is associated with symbiotic relationship Microbial community has an important impact on development of the Leishmania parasite in the host digestive tract. So it is believed that if the midgut re Leishmania jodi development hochi pro mastigote ru a mastigote, that means certain microbiota or microorganism present in the gut of the sandfly, they have a signifying role for the development of this pro mastigote. So this is the role played by the midgut microbiota in the sunfly. So paratransgenesis is a approach in which native bacterial flora of the disease transmitting vectors are engineered to interfere with the bacterial capacity. So এটা হচ্ছে কোন এই কোয়ালিটিটা যেটা তাଙ୍କର जो microbiota রয়েছে they help in the development of the leishmaniasis promastigote into ML. We can target this ability to prepare uh, where native bacteria flora of the disease transmitting vectors they are engineered মানে ତାକୁ transform କରିବା modify କରିବା genetical engineering method ରେ modify କରିବା. So they are called modified organisms ତାକୁ ସ୍ଥାପନ କରିବା ସେଠି, and they will not allow the development of this promastigate into a mastigate. So they will halt without killing the sandfly, they will halt the transmission of the parasite. Our aim is to that these parasite will get exhausted. They should be removed um, from the region, from the community. So one such method is that if we can engineer, if we can modify, okay, if you can transform these natural bacterial flora of the sandfly into a stage of the microflora which will interfere with the vectorial capacity. What is vectorial capacity? The capacity of the vector to sustain the load of the parasite and to provide a ample state so that they can multiply effectively for the next transmission to the next stage or the next animal. Now, pathogenicity, they cause kala azar, which is fever, and affects the internal organs. Reticuloendothelial system means it is present in the spleen, in the liver, and sometimes the presence of different pigments, they impart the skin a very dark color. And the visceral leishmaniasis, the cutaneous leishmaniasis, they bring about the nodulous, okay, nodulous granulous growth on the skin रे गोटे सब formation abu bholi formation which gives the skin a leprosy like lesions means growth, that is called cutaneous leishmaniasis. And mucocutaneous, they they bring about the development of the ulcer, mucosal ulcer in the nasal epithelium and the oral or in the mouth cavity. So these visceral leishmaniasis, the kala azar means kala jara, the black fever or the high fever. Okay, kala azar. So visceral leishmaniasis, the features is that weightlessness, then splenomegaly, hepatomegaly, increase in size of the liver, hepatomegaly, increase in size of the spleen, splenomegaly. These are the symptoms you see here. Okay. Then pancytopenia means lack of RBC, which leads to anemia, and lack of platelet, thrombocytopenia, leukopenia, WBC decreases. The skin becomes very dry, hands and feet and abdomen becomes dark. Okay, that is called. And so when the skin becomes dark along with the appearance of fever, it is called kala jara ba kala azar, black fever the skin becomes or black sickness. Okay, black sickness. The hair becomes brittle because brittleness janicho sahajare bhangi jibo. High total protein level and low albumin level present. Now, this cutaneous leishmaniasis, is associated with the cutaneous growth or painful ulceration in different parts of the body, which are exposed to sandfly bites. That means legs, arms, or face, and it reduces the victim's ability to work. So, ଘା ବଳି ହେଇଯିବ ତାଙ୍କ region ରେ. ଘା means ଦେଖିବ the periphery. Just let me see. Prepare କରୁଛି. Okay, let me see. You can so am I visible now?
Yes,
Yes,
sir.
sir.
Yes, sir.
Okay, I have some issue. I cannot resolve it in this Mac. Okay, in this Apple, I cannot
No
resolve the
problem,
issue.
sir.
Have to wait. Well, okay, cutaneous leishmaniasis स्किन ऊपर जो legs, arm, face जोटा expose होची जोटी sunfly bite करची सेटा घा बड़ी ହେଇଯିବ तार periphery घा जोटा ही जाउची the ulcer ही जाउची periphery becomes raised and this middle region it becomes an area for the secondary infection by different types of microorganisms or bacteria. So these are called ulcerative sores, and they can change in size and appearance over time, and they may evolve into. So simply, their origin occurs in the form of papules. So papules means घिमिरी type ro छोटो छोटो enlargement, छोटो eruptions. Then these become larger. That is called nodular. Nodular plaques to ulcerative lesions. Ulcerative means like high from which fluids come out, ulcerative lesions, with a raised border and central depression, which might be covered by crust. Crust mane hochi when it becomes dry, ta upare gote dry hard covering taku kaje crust covering. Some lesions persist as nodules, and uh, usually they are painless, but sometimes they become painful. Ketabole ଯେତେବେଳେ ulceration ହେଇକିରି it provides a medium for the infection by other bacteria ସେତିକି ବେଳେ painful ନହେଲେ sandfly ର ଯୋଉ ulceration ହବ that is less painful. Some people have swollen glands near the sores, for instance under the arm. Then mucocutaneous leishmaniasis ରେ ଆମେ କ'ଣ ପାଇବା ସେଥିରେ ପାଇବା ulcer similar to the oriental sore around the margin of the mouth and nose. छोट छोट nose ଭିତରେ ulceration the redness ହେଇଯିବ mucous area ରେ, mouth ଭିତରେ redness ହେଇଯିବ. Generally oral or pharyngeal symptoms sometimes are noticed, which are followed by persistent of abnormal nasal symptoms. Abnormal nasal symptom ହଉଛି କ'ଣ nose ଟା stuffing ହେଇଯିବ, blockage ହେଇଯିବ, difficulty in breathing ହବ, choking ହେଇଯିବ, stuffiness ହେଇଯିବ. So if this disease is in such state, if it is not treated, then there is a considerable destruction of the mucosal lining of naso-oropharyngeal mucosa, and the it may lead to perforation of the nasal septum. Nasal septum कोटा जो नॉस्ट्रिल रो दी भाग रही मझिरे जो बाट रही सेटाक भी से माने परफोरेशन ହେଇଯିବ परफोरेशन हे गले त माने ग्रॉस डिस्ट्रक्शन it may occur. So mucosal leishmaniasis usually become clinically evident within several years, sometimes decades of the original cutaneous lesion. So they may get transformed into mucocutaneous after several years. So ଆମେ fluid ବା blood को examine କଲେ ଆମେ ପାଇଯିବା leishmaniasis diagnosis କରିପାରିବା. investigate stage in the blood films. Smears are also can be seen. Just a minute. So they are stained with a Leishman or Giemsa stain, as per the name Leishmania Giemsa stain. तो blue जो pink color brinjal color जो ये रहुची सेतरे ताको ये करा जाउची. It is utilized. Then molecular diagnosis, that means through polymerase chain reaction. So joti polymerase chain reaction ame taro diagnosis kariba, it involves the DNA of that organism, and this DNA is amplified through polymerase chain reaction, so that the large amount we can study their particular DNA, on the basis of which we can make a remark that the person or the condition has been affected by or infected by. So antigen detection through ELISA, the presence of a specific antigen related to the Leishmania, which can be detected by ELISA test, enzyme-linked immunosorbent assay. Then there is another test, Leishmanin test. So immunity evasion mechanism, এটা same thing রয়েছে. immunity মানে লেশমেনিয়া প্রতি আমার বডিরে immune system রয়েছে, but যদি গোটে রোগ হଉଛି, if we suffer from, although we have immunity power to fight against different disease protein, so if at all we suffer from the disease, that means the organism have certain means to evade, okay, evade or to block or to mask the host immune response. সেটা আমরা এখানে পড়া যাচ্ছে, the immunity and immunity evasion mechanism, how will they fool the host immune system by different means. So host immune system, always there is a complementary system which aims at killing the promastigate forms in the bloodstream of the infected host. Then cells of innate immunity. The sunfly bite color, the first cells to reach the site are the neutrophils. So neutrophil, I mean, innate immunity means the immune system which we have inherited from birth. So from birth we have WBC, which are the protecting cells of our body. So these the neutrophils, they have the power of they are phagocytic in nature. ତାଙ୍କର ସଂଖ୍ୟା ନିଶ୍ଚିତ ଭାବରେ ସବୁ ସେଲ୍ ଠାରୁ ଅଧିକ 62%. So they engulf Leishmania promastigode. So if when the number of neutrophils is very large in comparison to the number of promastigode they have entered through the bite of the sunfly, but still then we sometimes the human beings suffer from the disease. What is the reason? That means they evade the killing by the neutrophil. ସେମାନେ କେମିତି ଗୋଟେ ସେଠାରୁ ମୁକ୍ତି ପାଇଯାନ୍ତି, by some means neutrophil ସେମାନେ ତାକୁ ଚିହ୍ନି ପାରେ ନାହିଁ. So subsequently during Leishmania infection, natural killer cells are also produced by the T lymphocytes. ସେମାନେ କ'ଣ କରିବେ, ତାକୁ kill କରିଦେବେ natural killer cells. they ତାକୁ କୁହାଯାଏ lysis, okay. So cytotoxic activity lysis କରିଦେବେ, ତାକୁ break down କରିଦେବେ, pores formation କରିଦେବେ ତା cell ରେ. तो छिद्र ହେଇଗଲା ପରେ water will enter and water entry ପରେ ସେ burst କରିଦେବ. So these are the ways by which the natural killer cells they kill them. Then cells of adaptive immunity, lymphocytes provide adaptive immune responses to Leishmania through the production of cytokines, which are the chemical messengers that are produced by the T cells. They play a major role in immunity in the various forms of Leishmania. So they stimulate or diminish the antiparasitic activity of the macrophages. So macrophages jota roichi, they diminish antiparasitic activity. Antiparasitic activity to stimulate karibo ba diminish kardeba antiparasitic activity of macrophage. Then macrophages which will come, which are produced by these reticuloendothelial cells or the spleen cells, semane they will kill these animals by phagocyting them. Um, so they also provide the lymphocyte. The leishmania ko habo, leishmania kichi mechanism bahar kartebo so that the macrophages ko tara anti engulfing power ta se kamai dabo. So it is called antiparasitic activity of the macrophages. ସେଇଥିପାଇଁ ତ ସେମାନେ they have the easy entry access to the human body or the body of the host and cause diseases. Then cytokines and chemokines, these are the inflammatory response by the immune cells during the infection of the leishmania, and this help in recruiting immune cells to the inflammatory site. So ये जो chemokines and cytokines ସେମାନେ release ହେଇଗଲା ପରେ immune cell को ସେ site of infection ମାନେ ଯୋଉଠି sandfly କାମୁଡିଛି ସେ ପାଖକୁ ସେମାନଙ୍କୁ ଡାକେ ମାନେ attract କରିବ, neutrophil ଯିବେ, macrophages ଯିବେ, and they are mediated by chemokines and the cytokines. In this way, the body immune system they prevent the attack of the leishmania. কিন্তু leishmania ର ମଧ୍ୟ immune evasion mechanism ରହିଛି by which it is responsible or it is capable to evoke the disease in the human body. तो ସେମାନେ ତାଙ୍କର modified complement system and phagocyte modified. So leishmania is able to evade the host. Evade माने host रो जो immune system, complement immune system जोटा जागृत होउची, जोटा activate होउची, ताकू से mask कर दबो, ताकू evade कर देबो using a variety of mechanism. Evade means it will cunningly escape that those complement system. ताकू कहे evasion. It will evade the phagocytic activity. So the modification in metacyclic leishmania elevated expression of protein kinase which deactivate complement pathways. So complement pathways re, there are several stages by which complement system is produced, and this any of these stages can be, if it is deactivated, the complement system will not be completed. Modified cell signaling ଆମର ସବୁ କିଛି ସବୁ activity ଗୁଡ଼ିକ cell signaling ର ହଉଛି. Cell will release certain signals which will attract in the form of chemokines. It will attract the neutrophils, it will attract the macrophages. ଯଦି cell signaling କୁ ଇଏ interfere କରିଲା, leishmania interferes with cell signaling cascade. Cascade ଅର୍ଥ ହଉଛି କ'ଣ? The entire process is by several stages. So ଗୋଟେ stage ହେଲା, that will allow next stage to occur, that will cause next stage. ଗୋଟିଏ stage ଯଦି ବନ୍ଦ ହେଇଗଲା ତାହେଲେ ପରବର୍ତ୍ତୀ stage ଆଉ ହବନି, ତାକୁ କୁହାଯାଏ cascade stage. Cascade involved in the synthesis and activation of macrophages. ତ ଇଏ ତାକୁ interfere କରିବ, ତାକୁ କରେଇ ଦବନି, ତାକୁ signaling କରେଇ ଦବନି. Then defective antigen presentation, ଆମର जो major histocompatibility complex ରହିଛି body ରେ, ସେମାନେ କ'ଣ କରନ୍ତି, they will carry the antigen and they will allow the T cells to notice the antigen, and then only the B cells will prepare the plasma cell, and this plasma cell will produce the antibody against the particular antigen. तो एटी कौन होची defective antigen presentation. Virulent stages of leishmania can regulate the expression of major histocompatibility complex molecules. से ताको प्रभावित करिवो, रेगुलेट माने कोन ताको चेंज कर दबो. तो चेंज हे गला पर सी आ प्रेजेंट करिवो नै. प्रेजेंट करिवा माने ता नियरर को नबा, का पाको नबा ना बी सेल पाको नबा, टी सेल लिम्फोसाइट पाको नबा, कोन नबा ना फ्रैगमेंट्स ऑफ द एंटीजन ऑफ द फॉरेन बॉडी. तो याको चेंज कर देला माने से माने चिनी परिबेनी. So that is why major molecules loaded with leishmania antigen or antigen presenting cell by interference with the loading. Then modified T cell element vaccine strategies against leishmania, since the approach kora chalchi, no ବର୍ତ୍ତମାନ full fledged vaccine ତିଆରି ହେଇ ପାରନି. So vaccine ତିଆରି ପାଇଁ they may take killed preparation ମାନେ attenuated. Killed होची totally, attenuated माने lifeless कर दबो. So killed preparation माने leishmania रो killed form रे ताको हमें utilize करिबा for the preparation of vaccine. Recombinant antigen vaccine recombinant DNA technology antigen vaccine तैयारी कर परिबा live attenuated parasite live माने parasite जीवित रहिछि किंतु एक्टिव नो इट विल नॉट मल्टीप्लाई ताकू कह जाय attenuated ता माने इनएक्टिव कर दिया गला killed आउ इनएक्टिव होछि टोटली डिफरेंट इनएक्टिव रो एक्टिव हे परिबो किंतु killed रो एक्टिव हे परिबो नाही. So among these, just vaccine based on attenuated parasite has shown efficacy. ता माने attenuated form जो तैयारी करा जाउची vaccine attenuated माने ताकू दुर्बल कर दिया गला तापरे ताकू यूटिलाइज करा गला फॉर वैक्सीन. सो एफिकेसी माने किछि परिमाण रो किछि देखा देइछि सम होप फॉर द डेवलपमेंट ऑफ वैक्सीन. देन the anti-leishmanial vaccines inactivated parasite administered by subjecting the parasite to antibiotics ताको जो attenuated करा गला inactivate करा गला केमती करा गला इति लिखिछि you can render them to antibiotics or chemical mutagenesis. chemical mutagenesis माने mutagen agent chemical agent दिया गला परे से mutation हे जाइतिबा तापर हीट द्वारा ताको अकमी कर दिया जाय परिबो रेडिएशन यूवी रेडिएशन द्वारा कमी कर दिया जाय परिबो. These are the different process by which you can produce the inactivated parasite or attenuated leishmania parasite which can be administered into the human body as a vaccine and it shows a certain degree of efficacy. So second generation vaccine subunit of vaccine composed of multiple single recombinant or native antigens तो मिश्रण करिकरी तैयारी करा जाय परिबो. DNA polyt of vaccines. So these are the different vaccines which are produced. सेटा तमे पढिबो. Vaccine कोन vaccine होची permanent therapy for not to allow the recurrence of leishmaniasis ताकू कजे vaccine. So vaccines it can be inoculated, it can preparation हे सरला परे ताको either orally दिया जाय परिबो नले subcutaneous दिया जाय परिबो नले skin रे दिया जाय परिबो. So anything we can frame a vaccine, peptide as vaccine, DNA vaccine, vaccine using salivary antigen of sandfly. तामाने sandfly रो जो saliva, so saliva सेटा भी foreign product रहिछि तार एंटीजन को नेकिर आमे तयारी करि परिबा. सो वैक्सीन सो दैट जेतबेले से saliva रिलीज करिवो so this antigen will not allow or it will bring about certain changes in the saliva जो थरे anticoagulant रहिछि और से changes रे these MST form will be killed. Now treatment होछि treatment माने सब drugs दिया जाउछि. So type of treatment depend करुछि को region रे the patients are present or the infective region ता ऊपर निर्भर करुछि. सब जगह रे सब प्रकार मेडिसिन दिया जाय नाही. liposomal amphotericin B is the recommended treatment in India. भारत वर्ष रे सेटा को यूज करा जाउछि often as a single dose गोटे थरे खाइबा को दिया जाय. Then pentavalent antimony compound दिया जाउछि but it has certain risk factors. Okay, risk factors. तापरे pentamidine isethionate is given to the patients at the rate 4 milligram. Anyway ए सब drugs रहिछि तो ड्रग्स भी सब बेले डेवलपमेंट रेजिस्टेंस डेवलपमेंट करूची लेशमानिया. तो रेजिस्टेंस काई करूची व्हेन गोटे प्रकार ड्रग जदी कंटीन्यूअसली यूज होबो अगेंस्ट ए पर्टिकुलर पैथोजेन ताले से पैथोजेन से ड्रग रेजिस्टेंट पावर इंक्रीज करि जेबो डेवलप करि जेबो. So nowadays many such drug molecules they are not सेटा भलो करंती नै डिजीज को. अर्लियर हमें कहू छे जे टेट्रासाइक्लिन हला कि आउ केते गुडा एंटीबायोटिक गुडी को से एबे इटा को यूज करा जाय पारुनी. New generation antibiotic, that means the particular bacteria they have developed the resistant factor against this antibiotic. The resistant factor होची just a change in their plasmid gene. The bacteria रे दी प्रकार क्रोमोसोम रोची तार मेटाबॉलिज्म पे गोटे मेन क्रोमोसोम रोची आ गोटे रोची एक्स्ट्रा क्रोमोसोम जेटा को प्लाज्मिड कोचे and this extra chromosome contains certain important extra characteristic coded in this gene. So one such coding gene होची resistant factor. So similarly जो लेशमानिया मध्ये drug resistant develop करी जाउची alteration in the active site of the drug without any alteration in the receptor. So possible mechanism of SAG drug resistance, clinical resistance to sodium antimony gluconate, SAG. A drugটা দিয়া যাଉଛି, so they have developed the resistance against this SAG. It is a major concern in the visceral leishmania treatment in India. So it can be attributed to the कौन ପାଇଁ से develop कला, प्रथम होची failure of drug inactivation caused by enzymatic conversion. Drug inactivate ହେଇ पारुनी, alteration in the active site of the drug without any alteration in the receptor. The drug active site, drug is a molecule, it will bind someone, ତାକୁ alter କରି ଦଉଛି ସେ without altering their receptor ଯୋଉଠି drug bind କରିବ. In this way they have developed the drug resistance. Possible mechanism of antimonial drug resistance ମଧ୍ୟ ସବୁ ପ୍ରକାର resistance ସେମାନେ क्रिएट करछंती, बट एग्जैक्ट मेकानिज्म कोथरे जଣା पड़ी नै. सब गुडी को प्रपोजल हाइपोथेटिकल दिया जाउची. सो वेरियस साइटेड रीजंस अल्टरेशन इन ड्रग सेंसिटिव साइट्स. तो जो रीजन ड्रग सेंसिटिव साइट माने सपोज गोटे ड्रग आमे गोटे बैक्टीरिया को ए ड्रग द्वारा बैक्टीरिया रोगटी भल हो जाउची. सो सपोज इफ दैट ड्रग इज एम्ड टू इंटरफियर with the formation of peptidoglycan which are necessary for the development of the cell wall. जदि से इंटरफेयर करिबो पेप्टिडोग्लाइकान हबनी, सेल वॉल फॉर्मेशन हबनी, बैक्टीरिया डेवलप करिबो नाही, बैक्टीरिया मल्टीप्लाई करिबो नाही. सेइथ में ताकू कोआ जाउची दे ड्रग सेंसिटिव साइट, को साइट ता मोस्टली ड्रग सेंसिटिव होची. तो सपोज गोटे तार रीजन लेशमेनिया ड्रग सेंसिटिव साइट, सपोज the drug aims at not to allow the development of the conversion of the flagellate stage into the non-flagellate stage. So seta site hochi, they will not interfere with the development of flagella, so that ta paravarti of development hobo nai sethi human being re. And if jodi drug tiyari kar dele se kon kala tar alter kar dela, alteration in drug sensitive site से सेटा को चेंज कर देला, आ गोटे साइड को हमें एम करिबा टू टारगेट आवर ड्रग. इन दिस वे टाइम गोज ऑन, बट इफेक्टिव ड्रग हमें डेवलप करि पारुनी. जहा डेवलप करि परछे ओनली फॉर कंट्रोल, नॉट फॉर प्रिवेंशन. तेनु प्रिवेंशन होची only to avoid the bite from the sandfly in sandfly prone area. ड्रग टार्गेट्स अगेंस्ट लेशमेनियासिस विभिन्न प्रकार टार्गेट मु जेमति कहितली को कहा को टार्गेट करिबा, एम करिबा फॉर द डेवलपमेंट ऑफ ड्रग, whether to aim for the development of their pellicle or the covering or the development of the flagella. So the different targets are there. Parasitic transporters, the transporters of these molecules, they can uptake toxic analogs of nucleosides that inhibit the cell growth of the parasite. So we can target their growth by interfering with their metabolism. এই উপায়ে বহুত গুড়ে টার্গেট এরিয়া রয়েছে যাহাকু লক্ষ্য করিকে আমি ড্রগ ডেভেলপমেন্ট করি পারিবা. আর প্রোফাইল্যাক্সিস হচ্ছে টু অ্যাভয়েড দি বাইট ফ্রম দি স্যান্ডফ্লাই দ্যাট উইল রিডিউস দি ইনফেকশন বাই লেশমেনিয়াসিস. সো নেট mosquito net use kariba ba net which is coated with insecticide taku use kari pariba. Insect repellent use kari pariba when we are moving. तो घर चारिपटे जो बुशी एरिया रही ताकू आमे इंसेक्टिसाइड दे करिबा, कट कर देबा, ताकू सब सफा कर देबा, और स्प्रेइंग ऑफ BHC और DDT as the insecticide. Cattle shed माने cattle region जो कैटल में रखुचे कैटल शेड गुडिक, इट शुड बी फ्रॉम ह्यूमन डिस्टेंस फ्रॉम ह्यूमन ड्वेलिंग. तापरे हाइजीनिक कंडीशन बिल्ड कले रोगटी व्यापिबो नाही. सो नेक्स्ट वी मूव ऑन टू द ट्राइपेनोसोमा gambians. So we'll chalk it out tomorrow, okay? let's discuss କିଛି ଯଦି କିଛି ଇଏ ରହିଛି doubts ରହିଛି students, do you have any doubt?
No, sir.
Shall we continue with the cystosoma or we will meet tomorrow?
Okay, sir. We will continue tomorrow, sir.
Okay, thank you.